The next study in this group is from Dr. Godler, and this is again is a renewal. So we had provided funding to Dr. Godler in previous cycles to look at what genes are differentially expressed both in blood and brain in individuals with PWS. So this is using human samples, and ask whether those gene expression differences are correlated with some of the behavior problems, the autism, the risk of mental health problems in PWS. And so Dr. Godler is looking at many things. He's comparing blood and brain in PWS compared to normal or typical, but is also comparing deletion to uniparental disomy to gain an understanding of why we see differences between those two subgroups. And he's done a lot of work on that and has, data sets about changes in gene expression across these different groups. And now we'll be, taking that to the next step. So in year 2, they're going to expand their studies to some additional samples. So they've done a number of brain samples, probably the largest group of PWS brain samples that's been investigated. They're going to expand that. And then they're looking in particular, they're interested in this gene UBE3A, which is in the PWS region. It appears to be expressed at higher levels in individuals with UPD and imprinting center mutations. And it appears that the higher it is, the more difficulty those individuals have, for example, with social cognition. So, the study will now expand samples and really, look at it in a larger sample set and correlate it with the severity of some of those behavioral changes. He's also going to use artificial intelligence to query those data sets and identify potential drugs that might improve these aspects of PWS. So we're excited about this study because it's been a very productive project in year 1. He's got very significant data sets. He's very collaborative. So again, it might be information that can be used across researchers and looking at the differences, again, between deletion and UPD. The application of new artificial intelligence approaches to identify drug candidates has been used in other fields, hasn't really been applied to PWS. So we're excited to see that applied here. And this is an experienced PWS researcher. So the goal here is to define the genetic pathways that are dysregulated in PWS blood and brain compared to typical individuals, as well as between the subgroups, and identify existing medications that could be used to correct some of these changes.